This is Masterstroke Education Biology classes. Today our topic biochemical reaction. To understand the biochemical reaction, we must first understand that what is a reaction. In a very simple word, all of we know that a reaction is a process where the substrate is converted into the product. So we can say easily A is converted to the B. That we call the reaction. Now A as converted to the B, so A is known as the substrate while B is the product. Now we are going to take some experiment to understand the reaction. However, the reaction is very simple process, but we are going to take undertake some simple experimental thought to understand the reaction. Think about it. In a condition 1, we take a glass of water. This is a glass of water. And we added some amount of sugar molecule in it. And put the glass in a table and leave it for 1 week or 1 month. After the 1 month, what we will observe? We are going to observe that the sugar molecule are completely dissolved in the water. This is the reaction. Very good. Now in the second phase, we take the same glass of water, we add some sugar molecule in it and we stir it with a glass rod, or you can choose anything. Okay, we stir it. What happened? The sugar molecule will be dissolved in the water, but the rate of the dissolve is much faster in the condition 2 than the type 1, because we are providing the additional energy by the stirring. In the number 3 condition, we take the water molecule, and we another time add sugar, and either we added HCl or we heat the glass or the solution. What happened? The sugar will dissolve in the water. So in the third phase, the water also dissolve in the water. So in each phase, we can observe that in each cases the sugar will dissolving in the water, but the rate of dissolve is much faster in the type 3 than the type 2 than the type 1. So what is the reaction? Reaction is a process where A is converted to the B, but reaction does not give us the knowledge that the rate of the reaction, is it fast or slow. In the fourth case, we take a glass of water molecule. Okay. Now we add some oil into it and we leave the water molecule like condition 1. We stir the solution with a glass rod like condition 2, and we heat or we provide the HCl like condition 3. What happened? In each cases, never the oil will mix with the water. So if a reaction will be occur, it will be occur, and if the reaction is not possible, so it will be never possible. So if A is converted to the B, it will be easily converted the substrate to product. But if there is no possibility that A will convert it to the B, so you cannot get bypass on it and you cannot convert A to B. So what here we can learn from this experiment, we are providing something externally, like here glass rod for stirring, here HCl or heat. These external forces that are responsible to accelerate the rate of the reaction, that are accelerated by the external forces. This is called the catalyst. Okay, this is called the catalyst. Now catalyst must be various type. Physical catalyst, such as in the example the glass rod, this is the physical catalyst. Heat, providing heat, it is the physical catalyst. Chemical catalyst, HCl, this is the chemical catalyst. The catalyst will never start or finish the reaction. The catalyst are only and only accelerate the rate of the reaction. For an example, we can see that if the substrate A will convert it to the product in presence of catalyst, think about 100 mole of substrate A will convert it to 100 mole of product B in presence of catalyst is required 1 minute, in absence of catalyst is required 60 minutes. So the thing is, A will be either or. In both cases they convert it to the B. But in presence of catalyst, the rate of the reaction is very fast. Okay, where it just required 1 minute to convert. Now, in living organism, from a single bacteria to a complex human cells, either it is a prokaryote, eukaryote, archaea, whatever in the taxonomic group, each and every living organism consisting some catalyst in its body or cell that are responsible to accelerate the rate of the living organism reaction. Such catalyst is known as the biocatalyst. So the biocatalysts are the enzymes present in our body, and HCl in our stomach is also known as the biocatalyst. Now the question is, every molecule or every substrate has a stable force. So why there is an environmental pressure upon the A? There is an environmental pressure upon the A, and it is a static force. It has a static force. Why A is going for the dynamic and converted to the B? So A must require to overcome its static force. Now, to understand this, we also going to understand exothermic, endothermic reaction and also the isothermic reaction. What is the isothermic? If the substrate and the product internal energy are same, this is called the isothermic reaction. But it is only theoretical. Practically there are no such reaction present that will be considered in biology, that will be considered as an isothermic reaction. What is the exothermic reaction? If A converted to the B and during the process heat released is known as the exothermic reaction. And the opposite, if the substrate A will convert it to the product B and the heat is gained by the product, this is called, the reactions require heat from the environment, this is called the endothermic reaction. But in detail, brief detail, we will going to understand what about this reaction. We are taking a graph here. This is the x-axis, this is the y-axis. Y-axis is for the energy. Okay, y-axis. Think about it. This is the 1, 2. This is the internal static energy of A, and this is the internal static energy of B. But the environmental suppressing energy, this is the environmental suppressing energy of the A is here. So this is the static energy. To convert into the B, A must need to absorb the energy from the environment to overcome the environmental energy. This position is called the transition state. This is a very important state of any reaction. Every substrate must reach the transition state. So up to this, what happened? A absorbing the energy from the environment. So up to this is the endothermic, right? It is absorbing. But the B internal energy is much lower than the A. So A must lose its extra energy and convert to the product B. So this phase is a exothermic. If the B static, if the C static energy is here, so A will absorb the environmental energy, produce in the transition stage, then gain another energy, and then that C. Okay. This is, so in this condition it is all the endothermic. So we can understand from this now what happened. During the reaction, we cannot say it is an endothermic reaction or exothermic reaction, because every reaction first half is an endothermic and the second third. So every reaction, the first half is always endothermic and the second half, it's depend, is it exothermic or it is endothermic. So only after the completion of the reaction we can say that it is an endothermic reaction or exothermic reaction by the mathematical calculation. But during the reaction we can never say it is the endothermic or exothermic reaction. So this is the A and B, this is the substrate and product we are talking about. So what is the role of the biocatalyst, catalyst or enzyme in it? So why will be enzyme interact? There are a lot of things we know. There is a Fisher lock and key model. There is a enzyme substrate specificity model. Enzyme has a catalytic subunit, allosteric subunit, enzyme regulatory subunit. There are a lot of things about the enzyme. But ultimately, the question is, why enzyme will be interact with the substrate? It is a very simple question, that it is a catalytic subunit for the substrate, it is a lock and key features for the substrate, so it will be interact. But okay, that good. But for the point of physics, the enzyme has its own static energy and environmental pressure. So why its catalytic unit will be interact with the substrate? Because the enzyme to interact with the substrate, the catalytic unit or catalytic site or the active site of the enzyme, if need to interact with the substrate, the enzyme also must overcome its environmental stage. So enzyme must need to overcome the environmental state, and then it bind with the substrate and push and help the substance to easily reach the transition stage. From this point we can understand that, think about it, we can, in this graph we can understand that every substrate must reach the transition stage, okay, to complete the reaction. Enzyme only and only help the substrate to reach the transition state in a faster, so the rate of the reaction is accelerated. Okay, without the enzyme, the substrate will be also in such condition overcome the transition stage, but it's required time. But in presence of the catalyst, biocatalyst or enzyme, the transition stage of the substrate will be easily reached. Okay, so this is all about the enzyme, biocatalyst and the reaction. Now, why we are talking so many things about it? Because in the biochemistry, we must understand the term the metabolism. What is metabolism? Metabolism means the processing of the macro and micro molecule. That is called the metabolism. The metabolism is actually divided into two major phases. One is called anabolism and another is called catabolism. Anabolism is also called growth processing, by which energy stored in the body and body growth will be take place. For an example, in case of the plant, photosynthesis is a very good example of the anabolism. For a human body, glycogenesis, by which glycogen is being synthesized in our body, is called the anabolism. In the opposite world, catabolism is a process where the stored food material are processed to burn and provide the energy to perform the bodily functions and to fill the demand. Okay? The catabolism good example is the glycolysis. However, the plant C2 cycle, in case of human or eukaryote glycolysis, beta oxidation of the fatty acid are also a very good example. Glycogenolysis are also a very good example of the metabolism. So metabolism is a process where the macro and micromolecule are processed. Okay, the metabolism divided into two parts, anabolism, catabolism. Anabolism is a growth related processing, where catabolism is food material breakdown and energy providing. That means just opposite of the anabolistic process. So the regulation between the two processes is very important. Every cell must need anabolism and catabolism process, and the stability or balance between them are very important. Okay. Over anabolism, for a human, think about it, the catabolism rate is much higher than the anabolism. Then there must be increased chances of the poor nutritional case, such as kwashiorkor marasmus or whatever. Such as in case of sugar patient, the catabolism rate, you know that the sugar will be immediately burned unnecessarily. So in case of the sugar or diabetes patient, the catabolism rate is much higher than the anabolism. And in case of the obesity cases, the anabolism is much higher than the catabolism. So the balance between them for the healthy bodies must be required. If the balance will be disbalanced, so what happened? Either cases, the clinical condition can be observed. Okay, this is all about the biochemical reaction, its type, its graphical representation, about the biocatalyst, catalyst and enzyme, and it is all about the metabolism and its different part, example and applications, and clinical approaches are also we discussed in this video. If you like the video, please like and subscribe our channel. Thank you.